Hello, everybody. My name is Lucia Ramirez-Joseph, and under the mentorship of Dr. Terui and Dr. Kim, I developed this research. So I'm looking into biotic and abiotic factors that affect seasonal patterns of fish body growth at an urban stream in the Piedmont region in North Carolina. So I am looking at fish, and I'm looking at their fish growth, their body growth. Why? Because fish are a pretty important aspect of our streams. They are everywhere, and we can find them pretty easily. Their body growth can tell us a lot about ecological characteristics like their health, their survival, their competitive ability, and there are different factors that can influence each individual's growth. So fish growth, and especially in streams, varies greatly depending on many things, which makes this an excellent study system. It's pretty cool to investigate and to go out and on the field and check for stuff. So the fish growth, like I had said before, can be affected by many things. For example, it can be affected by competition, or which is the biotic factor, or an abiotic factor like habitat size and temperature. So for my research, I looked into how fish density and the season affect the growth rate of recaptured fish in a specific stream. So for this specific stream, we went to the UNCG Gateway Research Park to the North Campus Creek. It's around 430 meters long, around 3 meters wide, though it varies sometimes. It has a mixed canopy, and their habitats also vary between pools and riffles. So we looked into 4 species of fish. 2 of them were sunfish. One was green sunfish, like you can see on the bottom left, and one was redbreast sunfish, like we can see on the bottom right. Our other 2 species were minnows, like bluehead chub on the bottom left and creek chub on the bottom right. So for sampling, we did 6 occasions of fish sampling. One was done in November in 2020, 4 of them were done in 2021, and then 1 was done in 2022. We always went February, May, August, November, and then repeat. So each time we went out, we went through 43 sections, or all of them were around 10 meters each, and we used an electroshocker to catch the fish. An electroshocker is like what you can see in my backpack over here in the picture. So for fish tagging, after we caught them, we identified them, we measured them in length and weight, and then we tagged them. If they were not tagged before, if they were a recapture, we would identify them again, measure them and weigh them again, and then let them loose. So for tagging, we use 12 millimeter HDX pit tags, and we used either scalpels or injectors to insert the tag depending on their size. We only tagged fish that were bigger than 60 millimeters. If they were smaller than that, we would record their species and record their total length. So for my study, we only used recaptured fish since we need to see the body growth, so we would need more than 1 occasion of catching them. So we were able to have 173 species of fish. Some of them were different. We had some more of some than the others, but this is what our sample looked like. So for our statistical analysis, we did linear regression model in R, and we were looking at the individual body length and their growth as a response variable. And their explanatory variable would be the population density and the temperature in months, which can also be looked at as like season. So results. This is a very complicated row plot looking at the density of the populations of fish versus the specific growth rate of the fishes, of the individuals. So we weren't able to find a lot of significant relations between most of them, but we did find 2 significant relations that I will talk about in a second. So I want to explain a little bit more. Here we can see the density. So all of these are the densities of the redbreast sunfish. And then here are the actual individuals. We can see how each were affected by the other species. So we can see bluehead versus bluehead versus creek chub, et cetera, for all of them, which is why this looks so dense and complicated. But when looking at it a little bit closer, like I had said, we couldn't find almost any significant relations between them. There was almost nothing. But we did have these 2, creek chub versus creek chub and green sunfish versus creek chub. So for creek chub versus creek chub, we found that the growth rate decreases with the population density of itself. So as their own creek chub are getting, the density is getting bigger, their growth rates are getting smaller. On the other hand, for the green sunfish, they are affected by a different species. They are affected by the creek chubs. So as the creek chubs get bigger, as their density gets big and their population, their growth rate gets smaller and smaller. So as the creek chubs population gets bigger, the green sunfish start to get smaller in size. This is just a relation in between that has not told us why this is happening or if there's any cause and effect here. So lastly, for our seasonality aspect of this research, we were looking into our months of recapture down here in the x-axis versus the specific growth rate of fish. And we found that there's a pretty clear pattern between all of them in all of the species that we were looking at. Between the colder months we found almost no growth in the fish. However, in the warm months, we found a lot of growth in all of the species. So this is a pretty specific pattern. When it's warm, the fish grow. So in conclusion, we found intraspecific density is affected by creek chub's growth. We found an interaction between green sunfish and creek chub. We don't know specifically what is causing this. We know that there is a relation between those two. We don't know if they're being affected by the same biotic or abiotic factor. We don't know which specific factor is causing this. We do know that there is something that is causing these fish to have a relation and to be decreasing in their growth rate. And finally, we also found that the fish grow during warm months, which is, like I said, a natural physiological response. So for future research, we could do probably a lot of things. This is like a good baseline to go from here. We could look into which specific factors are affecting the growth of green sunfish and the creek chubs. Like I said before, since we don't know, it could be a cool thing to find out what is affecting this growth rate. And then we can also look into why the density is not influential for most pairs of species. Like I showed in the big dense row plot, a lot of that, out of all the 16 scatter plots, only 2 of them were significant. So we could find out why the other ones were not significant. Like what is happening that is that stream really good in resources and that way there is no competition between them, so nobody is being affected, or what is happening? And we could also look into the different microhabitats. We could zoom in into something and focus on that. So that is all for me today. Thank you if you came this far. I will try to answer any questions you have. Thank you so much.